The research labs of the Transplantation Institute all work for the same goal, and that is to eventually have no patients on the waiting list. Fundamental and translational research is done for all organs that are transplanted in the Erasmus MC, which are lungs, heart, kidney and liver. Me and my colleague researchers aim to get better insight into end-stage organ disease, how to better preserve organs, study organ regeneration, including tissue engineering, and dive into transplantation immunology and related pathology. Now, when people think about a lab, this kind of steampunk-like images arise in their minds. Brass instruments, bubbling concoctions in an atmosphere where imagination meets invention. And where nutty researchers come to life, complete with quirky props and eccentric charm. Well, the reality is slightly different, I must admit. Our labs are cutting edge, with modern equipment and brilliant, yet less nutty researchers. Today I will set liver-related research as an example, since that is what I know most about. But similar type research is done for the other transplant organs in our institute, including the combinations of organs. We use several techniques to make organ models, so-called liver models in a dish, which means that we try to mimic the disease and transplantation-related issues. Amongst other technology, we use three-dimensional cell cultures. These human so-called mini-livers are named organoids. Even more complex is when we add an artificial blood flow in a liver or chip or 3D printing models. We furthermore use a technique named decellularization and recellularization to tissue engineer functional liver tissue in the lab and use machine perfusion technology, which is already used in the clinic during transplantation procedures, to repair organs. In the next few slides, I will show you some examples. So let's dive into the world of organoids, or mini-livers, and how can we use these cultures to model liver diseases? What are organoids? Well, we start with a small liver biopsy, and with a special procedure we grow 3D cell cultures of these balls or spheroids called organoids. Each ball consists of thousands of cells that self-organize in these spherical structures. And particularly liver-derived organoids, they grow pretty fast. Within 6 days, they have grown from single cells to an organoid of thousands of cells. So with these organoids, we can model diseases. For instance, when using liver biopsies of patients with a disease such as genetic enzyme deficiencies. We can also infect organoids with viruses such as hepatitis viruses and SARS-CoV-1. We can model many liver-related diseases in the lab. And in these pictures we show proof that the organoids mimic the original disease as seen in the patient. For this we use many techniques such as imaging and gene expression profiling. Another very exciting research path is to use machine perfusion. This machine is already used in the clinic to assess the organ quality before transplantation, but it can also be used in an experimental setting, for instance to study liver regeneration. It also provides a great opportunity to do therapeutic interventions. While being outside the donor and not yet transplanted inside the patient, cells or specific drugs can be added to repair an organ or to remove fat in case of a liver that has too much fat to be safely transplanted. Again, here is an example of stem cell therapy during machine perfusion, and of course a lot of analysis after the procedure needs to be done to see if and how this worked and what can we learn from the experiment. The final technique I want to share with you today is decellularization of organs that are not suited for transplantation. So instead of discarding these organs, we try to use it for tissue engineering and making functional liver tissue in the lab. And yes, sorry again to be using the liver as an example. Here a liver, in this case a pig liver, of which we remove all cells via perfusion in this setup. You can see that the cells are rapidly removed and transparent spots become visible. And we end up with only the scaffold, No cells are there anymore. And with this we can now study what the scaffold looks like, what proteins are in there, but also use it as a basis for tissue engineering. Here is a schematic representation of the process of decellularization and recellularization. As seen here, we infuse new and functional cells in the, in this case human, decellularized liver again. Many other techniques and research projects are ongoing in the laboratories of the Transplant Institute. I just highlighted a few. And pretty biased, of course, only showing liver. If you are interested, just contact us through the Transplant Institute. You are more than welcome to come and have a look in our labs and see the other cool stuff we are doing. Thank you for watching.